This is the component 1 in class task book for the upper 6 and question 3. This is a classic experiment. If you look at old biology books and questions, they'll always have this in. Respiration in maggots over a 5-minute time period. We've got 5 cm cubed of sodium hydroxide solution, and that absorbs CO2. So any CO2 that's being produced by these maggots will get absorbed and taken away. It's corrosive. That's going to help us in the question. And then they open the tap up, and we've got a capillary tube with a bubble in there, and presumably the bubble's going to move one way or other in this experiment. And we are leaving this to start with for 10 minutes. It's in a water bath. Okay, and then we are doing another experiment using water instead of this sodium hydroxide solution. Okay. So what's going to happen is these maggots are going to respire. They're going to use up glucose. They're going to use up oxygen, and they're going to produce CO2 and water. They're going to use up oxygen out of the air and produce CO2, but the CO2 is going to go into the sodium hydroxide solution. So oxygen in, CO2 into the solution, so this bubble would move to reflect the change in the pressure when the tap was closed. So when we close this tap, oxygen is used up, this bubble moves along, and because we know the size of the tube and how far it's moved on, we can calculate the volume of oxygen produced. Right, why they're placed in the wire cage? Well, we don't want these little critters falling into the sodium hydroxide solution because it's corrosive. So maggots do not contact the NaOH, or possibly even drown or drown. Either way, they won't like that. There are 2 reasons why the apparatus was left in the water bath. So what they're putting this entire setup in the water bath at 25 degrees C. As soon as you put it in the water bath, these gases in there will expand, so this bubble will move away if the tap's closed. And also these maggots, who previously might have been at 10 degrees, 10 or 15 degrees possibly, then they need to adjust to the new temperature or acclimatise to the new temperature. So just 2 reasons here. So we'll have give time for the maggots to acclimatise. Gives time for the maggots to acclimatise or get used to these new temperature conditions. And we also mentioned that as soon as you put this tube in a water bath, the gases will expand, and that'll affect where the bubble is. So gases may expand, presumably if we're heating them up. Another thing you could possibly say, gives time for the solutions to equilibrate to the right temperature. If that was going to make a difference, it won't really make a difference, but it's another point we can say. So time for NaOH to equilibrate to the new temperature. Suggest a suitable control. So if this experiment is trying to find out what the maggots are doing, and that's affecting where the bubble is going to be. Well, the control is, let's do it without the maggots. In fact, let's replace the maggots with something else. With plants, we'd usually boil it and cool it, but probably not the fairest thing to do to the maggots. So we're going to replace them with glass beads or dead maggots, if we can still do that. And that would show that the changes in the volumes that we're measuring are due to the maggots and not anything else. So 2 marks there. One is going to be what we're actually going to do, and the second is why. So carry out the same experiment, but using dead maggots, or possibly glass beads if you don't want to kill the maggots. That would show that it is the respiration of maggots causing the volume changes. So pretty straightforward, typical question so far. Here's our results. When we've got water there, nothing seems to happen. That's probably because instead of the CO2 going down into this liquid and being absorbed, the CO2 just replaces the oxygen. So we're using a volume of oxygen, we're producing a volume of CO2, and so the overall pressure will be the same. So that's why the water doesn't have any movement. And we've got movement with the sodium hydroxide. So we just need to explain why the coloured liquid moved but did not move in experiment 2. So we're just going to summarise that. So experiment 1, and that's with the sodium hydroxide. The oxygen is used by the maggots in aerobic respiration. O2 used by maggots in aerobic respiration. And why does it move? Because this CO2 is being absorbed by the sodium hydroxide. So the CO2 released by the maggots, CO2 released by the maggots, and it's important to say it was the CO2 that the maggots produced in respiration, absorbed by the sodium hydroxide. And so the volume decreases in the tube. So it will decrease the pressure, and so that bubble will move. So volume will decrease, and that moves the position of the coloured liquid. And experiment 2, which is water, the volume of CO2 produced equals the volume of oxygen used up, so the overall volume is equal. So volume of O2 used equal to volume of CO2 released. And explain the advantages of collecting the results from the whole class. So this is, you might have put this in your lab books when talking about why you do repeat results. The answer is always the same. it increases the reliability and reduces the effect of anomalous results when you calculate a mean. So increases reliability. Increases reliability of the mean that you produce. This would reduce the effect of anomalous results. And we could, now that we've got some repeats, we could start doing stats on this. So it allows us to use a statistical test. So now allows the use of a statistical test if we were comparing the means in a t-test or something like that. And here's a nice little calculation. We've got the diameter of this tube, massive maggots, and key bits of information. So that's the diameter of the tube, that's the mass of maggots. We've got to do this for 5 minutes. We know what pi is. What do they ask us to calculate? Millimeter cubed per gram per minute. Right, we're going to get our millimeter cubed from our result, which was in 5 minutes, that was 61 millimeters. So this is that has moved 61 millimeters, and we know what this size of tube is. That is diameter is 1.2, radius equals 0.6. These maggots, they weigh 0.5 grams. So diameter was, so radius equals 0.6. We have got our 61 millimeters. So we've got all the information now. So the rate, we need to do this, calculate this first. So we've got pi r squared h, so 3.14 times by r, which is 0.6, times by 0.6, which is r squared, times by 61. And then we want that per minute, and we've got 5 of those, and we want it per gram, and we've got 0.5. So we're essentially going to have to divide our answer by 5 to get it per minute, and then times it by 2 to get it per gram. So we're going to divide that by 5 and then divide that again by 0.5. So let's try that. I hope this works. So 3.14 times by 0.6 times by 0.6 times by 61, and we're going to go divide by our 5 minutes and then divide by our 0.5 grams, or times it by 2, same thing. And that's our answer, 27.58176. And finally it goes, how are we going to give our answer to 1 decimal place? So it's going to be 27.1 decimal place, that'll be, next one's 8, so that'll be 0.6, so 27.6, and that's our answer. Phew. So pretty involved calculation there. We've got this crazy equation, respiratory quotient, and a bit of blurb to read there. It's CO2 over O2. And when you do that for your normal equation, we got 6 CO2 divided by 6 O2. So 6 divided by 6 is 1. And calculate the RQ for tripalmitin. So what have we got here? It'll be CO2, which is 1O2. And how many oxygens have we got? 145. So the RQ is CO2 divided by O2. 1O2 divided by 145. 1O2 divided by 145. And that's 0.703. Does it tell us what decimal place is? No, it doesn't. So I'm going to put 0.7. It doesn't tell us. Use the equation for respiration as tripalmitin. Suggests why desert animals such as camels use fat as a substrate for respiration. So 2 marks. So this is all about metabolic water. So desert camels, well, why are they doing this in the first place? There's a lack of water in their environment. Not enough just to say it's hot. You need to talk about lack of water, and this essentially produces more water. So when you work this out, you got, if you look at the how much substrate and oxygen you've got producing how much water, well, when you do it this way, you're producing a lot more water, and this is our metabolic water. And so camels, when they say the water's in the hump, well, you're nearly right. It's a big mass of fat in the hump, but it will produce lots of metabolic water when it's metabolized and respired. So the hump does lead to water. So this produces a lot of metabolic water from respiration. And that's the end of that.